Hello, today we're going to explore the brain. Here we see an actual picture of the outer surface of the brain. We can see the convoluted areas that make up the outer layer of the brain called the cortex. We can see the vessels that supply blood to our brain. Our brain runs on oxygen, which is supplied by blood, and also glycogen, which is also supplied through the circulatory system, and our brain is a large consumer of both of glycogen and oxygen. One of the exciting developments in recent years in studying the brain is in the area of neuroscience, where various kinds of methods are now used to look at what's going on in the brain. One of these is an electroencephalograph or EEG machine. These have been around for quite a while, and they record the electrical activity of the brain. When we get into our discussion about sleep, we'll see how the EEG machine has helped researchers to differentiate different stages of sleep. You may have some familiarity with PET scans, which are another way of looking at what's going on in the brain. And we're going to see a picture of a PET scan here in just a moment. MRI machines and what are called functional MRI machines are also used to study what's going on in the brain. The fMRIs actually look at what's happening when somebody is doing something, which is a pretty cool way of looking at actual brain activity when it's occurring. I have some neuroimaging pictures I'd like to share with you. You will not find these in your chapter, but I think they're interesting to look at. Here we see an MRI of a large tumor. It's quite obvious where the tumor is located in this person's brain. This would be in the temporal lobe area of the brain. It's a fairly good sized tumor. Here we see a CAT scan showing a mass of blood within the brain. We can tell that the blood is putting pressure on the brain, because this section right here should look sort of like a wishbone or a V, and it should be moved over to the very center. It's called a ventricle. It's one of the fluid-filled areas of the brain. You can see it's displaced over because of pressure being exerted from a mass of blood within the brain. Here we see a PET scan showing a normal brain, a person with mild cognitive impairment, and someone with Alzheimer's disease. We were looking earlier at how the brain runs on glycogen and oxygen. You can inject a sort of a tracer into the bloodstream of a person that lets you see the parts of the brain that are most active in terms of their utilization of glycogen. And when the brain is relatively more active, more glycogen is being consumed. So starting with red, we can go from red being the most active down to orange, to yellow, to green, to blue, to black. So in looking at these PET scans, the most active areas are red, and the least active or inactive areas would be black. So you can see how when someone has mild cognitive impairment, there are areas of the brain that are not as active in a normal brain, and then a rather dramatic decrease in activity levels in different areas of the brain for somebody with Alzheimer's disease. Here we see a comparison of PET scans of someone who has a normal brain and someone who is a methamphetamine abuser 1 month after detox and 24 months after detox. This area of the brain that we see is the brain's reward pleasure center. It has been found that when you or I experience normal pleasurable activities, there's an increase in the production of dopamine in our brain's reward pleasure centers. When someone abuses drugs like methamphetamine, it causes a rush of dopamine in their brain's reward pleasure areas. And eventually it depletes the dopamine neurons so they cannot produce dopamine in the normal way, causing the person to have to rely on the drug in order to experience pleasure. As you can see, for the meth abuser 1 month into detox meaning they're getting off or are now off the drug, there's less red, meaning less dopamine activity going on. But 24 months after detox, you can see that that red area is coming back, meaning those dopamine neurons are starting to function more normally again, which of course is a good sign. Here we see the brain of someone who's normal compared to someone with obsessive compulsive disorder. You can see this area of the brain, which is the frontal lobe of the brain, is very active in a person with OCD. The frontal lobe area of the brain is the thinking area of our brain. And someone with OCD overthinks. They have obsessive thoughts that they can't rid their mind of. So this part of this person's brain is lit up like a Christmas tree. It's overactive. Here we see a comparison of the brain of a healthy individual on this side and someone with schizophrenia on facing the picture would be on your right. And you can see one difference in these two scans is this area here is smaller than this area here. This is the ventricle area. A larger ventricle area here means that probably there's, for whatever reason, death of neurons in this area, allowing this space to be larger than in someone with a normal brain. Let's take a look at some of the structures of the brain. In thinking about the organization of the brain, different structures in the brain and their functions, we can think of the lower areas of the brain as being more the more primitive areas of the brain, and the higher areas of the brain being the more evolved areas. So at the base of our brain, as you can see in this picture, we have the medulla, the pons, which make up the brainstem, the reticular formation, and then the thalamus. And you can see them here relative to other areas of the brain, structures of the brain. So the medulla is the part of our brain that regulates vital functions like breathing and respiration. And the pons helps to coordinate movement and also to control sleep. And along with the reticular formation, this whole area here helps to control arousal and sends pathways or fibers up into the higher areas of the brain that help us to regulate our level of alertness or arousal. The thalamus, as you can see, sitting atop this area, is like a sensory relay station in the brain. Sensory inputs coming in from different areas go through the thalamus before they then end up in the part of the brain that registers that particular sensation. Another interesting area of our brain is the cerebellum, As you can see, it's at the base of the brain, about the size of a baseball. And it helps to regulate coordination. And as we'll see later, it's also involved in the storing of unconscious memories involving motor memories and also memories learned through a kind of learning called classical conditioning. Another area of our brain of great interest to psychologists is the limbic system. The limbic system includes the hypothalamus, the amygdala, and the hippocampus. You can see the pituitary gland in this picture as well, but please remember the pituitary gland is actually a part of the endocrine system, not the nervous system. And it is regulated by the hypothalamus, a part of the nervous system right above it. The hypothalamus helps to regulate internal motivational states of our body for things like hunger, thirst, sex. The hypothalamus, as I said, also regulates the activity of the pituitary gland, which regulates other glands of the body. Hypothalamus helps to regulate the internal environment of the body to help us to stay comfortable. And if things get out of balance, then our homeostasis is disrupted, and the hypothalamus tries to restore that. Here you can see the thalamus, which we talked about earlier. This is the hippocampus, which I mentioned in an earlier discussion when we were talking about Alzheimer's disease. The hippocampus is very important in helping to temporarily store certain kinds of memories. They're not permanently stored there. And these memories are processed and then sent on to the cortex of the brain, where they are permanently stored. The amygdala is often referred to as the emotional processor of the brain. It becomes involved in the experiencing of different emotional states. In this picture, we can see a three-dimensional view of the limbic system and other structures of the brain. Another structure that's mentioned in this picture is the corpus callosum. As you can see it here, the corpus callosum helps the two halves of the brain, the two hemispheres of the brain, to communicate with one another. We'll be returning to the corpus callosum a little later. As you are probably aware, there's been a lot of interest in recent years in how athletes may experience brain damage resulting from injuries to their brain and concussions. There's an interesting study mentioned in our chapter where they looked at the brains of 111 deceased NFL players and found 99% showed signs of degeneration related to frequent head trauma. As I'm sure you're aware, these kinds of research findings have brought about some changes in the rules of football and in the equipment that football players use. But players of other sports can also experience concussions, and we may well have students in our class who have experienced a concussion at some time, or maybe even more than one, at some time in their lives through sports, or perhaps because of some other kind of injury. Our brain is organized into two hemispheres, and each hemisphere is organized into 4 lobes: frontal, parietal, temporal, and occipital lobes. We're going to be talking some about each of these hemispheres and their functions. As we look at the two hemispheres, we'll be looking at the 4 lobes of the brain, and their specialized functions. Here we can see the frontal lobe, the temporal lobe, the parietal lobe, and the occipital lobe. This is the cerebellum, this is the spinal cord. At the very back of our frontal lobe is what's called the motor cortex. The cortex is the outer layer of the brain. On the motor cortex, our body is laid out in such a way that those parts of our body that are the most needed for movement have more innervation or surface area on the motor cortex, as you can see in this diagram here, also called a homunculus. And then at the very front of the parietal lobe is what's called the somatosensory cortex. And again, our body is laid out on this area proportional to how important these different parts of our brain are for innervating parts of our body that are very sensitive to touch. The visual cortex in the occipital lobe is the area of our brain that allows us to experience vision, and the auditory cortex in the temporal lobe allows us to experience hearing. We also have areas in our brain called association areas. These are involved in higher mental functions like learning, thinking, remembering, speaking. One area of our brain that is rich in association areas is the frontal lobe of our brain. And the frontal lobe of our brain includes the prefrontal cortex, which is the front of the frontal lobe. And this area of our brain allows us to experience judgment, planning, to process new memories. It has so many different important functions. And damage to this area can, among other things, interfere with our ability to think and plan ahead. You may be aware of the famous case of Phineas Gage, who experienced a work-related injury back in the 1800s when an explosion caused a rod to completely travel through his brain and land a number of yards away. You can see Phineas holding the rod in this picture. The injury caused damage to his left frontal lobe and resulted in a dramatic change in his personality to where before the accident he was an easygoing, mild-mannered man. After the accident, he became very explosive, angry, short-tempered, and was very difficult to get along with. This case of Phineas Gage, the most famous case in all of neuroscience, among other things, helped neuroscientists to realize that how important the frontal lobe area of our brain is to regulating our personality. We also have areas in our brain associated with language and speech. One of these is Broca's area in the frontal lobe of brain and Wernicke's area in the temporal lobe area of the brain. Broca's area helps us to be able to say words, and Wernicke's area helps us to be able to understand what is being said to us and to put words together in sentences that make sense. If someone experiences, say, a stroke that damages one or both of these areas of the brain, they will experience aphasia that can affect their ability to express words and also to understand words, depending upon the extent and location of the damage. We started out our discussion of this chapter looking at the idea of plasticity, I'd like to return to it again just for a moment. Looking at this phenomenon where brain damage sometimes is compensated for by the brain when surrounding neurons that are not damaged form new connections that help to at least in part result in some restoration of function that may have been lost because of the damage. There is some formation of new neurons in the brain through the process of neurogenesis. And one interesting area of research that's being explored nowadays is to take stem cells, which are new undifferentiated cells, and implant them in different areas of the brain so that they can then take on the characteristics of the neurons in that area and help that part of the brain to function better. We mentioned earlier that there is a part of the brain called the corpus callosum. Here is the corpus callosum. It extends outward. You can see it here in this cool picture. And it connects the two hemispheres and helps them to communicate with one another. Occasionally, the corpus callosum is severed intentionally through surgery as a way of controlling epileptic seizures. If someone experiences epilepsy, there's kind of an electrical storm that occurs in one area of the brain, and then it can spread and it can be transferred from one hemisphere to the other. Well, if you sever the corpus callosum, it helps to control the spread of the seizure activity. It's not life-threatening, but it, of course, does affect the person's functioning in some ways. They learn to live with it, and it doesn't have to really disrupt their life all that much. But on the good side, of course, it can help to control their seizures, hopefully. Researchers have studied how the two hemispheres of the brain communicate with one another using patients who have had split brain surgery where their corpus callosum has been severed. Taking this diagram as an example, something that appears in your left visual field eventually shows up in your right occipital cortex, whereas something that appears in your right visual field eventually shows up in your left occipital lobe, where your visual cortex is, because of course the left side of the body is controlled primarily by the right hemisphere of the brain, and the right side of the body is controlled by the left hemisphere of the brain. Well, if a person's corpus callosum is intact and they are, say, looking into an apparatus, kind of like what you look through when you go to the eye doctor, where you're focusing on your chin is stationary, you're focusing on a dot in the very middle of your visual field. If a pencil were to flash in your left visual field, it would register back here in your right occipital lobe. If the apple were to flash in your right visual field, it would register back here in your left occipital lobe in the visual cortex. So if the experimenter asked what you just saw when the pencil was presented, you would be able to say a pencil. And if the researcher asked, what did you just see when the apple is presented, you would be able to say an apple. But what if the corpus callosum were severed? Then what would you say? The picture is presented in your left visual field. It registers in your right occipital lobe. And if the researcher says, what did you just see? you would say nothing, because you wouldn't be able to communicate to the speech area of your left hemisphere, which is where the speech center is located in the left hemisphere, what you just saw. It registered, but you couldn't acknowledge that you saw it because it didn't get over to this side of your brain. If they were to give you a pencil and ask, can you draw something that you just saw, you'd be able to draw the pencil and then you can label it, but you wouldn't be able to say what it was to start with. You might say, I don't know, or nothing, or something like that. But if the researcher asked what you saw when they presented the apple in your right visual field, which registers in your left occipital lobe, you would be able to say an apple because it's in your left hemisphere primarily where our speech centers, Broca's area and Wernicke's area, are located. So that part of your brain is communicating within itself, but it can't communicate with the right hemisphere. Here's another example of what we're talking about with split brain. The subject is asked to look at the dot, and then the letters HE and ART appear. HE registers in the right occipital area. ART registers in the left occipital area. The person is asked, what did you see? And they'll say art, because art registered in the left occipital area where their speech centers are. And if the person were, say, asked to point with their left hand to the word they saw, they would point to he, because that registered in their right occipital area. In thinking about the two halves or hemispheres of the brain and how they communicate with one another by way of the corpus callosum, we might also consider some differences in functioning of the left and right hemispheres. As you can see, each hemisphere performs distinct functions. The left hemisphere is more the logical hemisphere of our brain, and the right hemisphere is the more sort of intuitive, creative part of our brain, which excels in making inferences or reasoned conclusions, whereas the left hemisphere is good at making quick, exact interpretations of language. This completes this part of our discussion about the biology of behavior and consciousness. Up next, in our next discussion, we'll be discussing consciousness. Thank you.